Hello, this is Matthew Wakeling. This is just a quick video to show how paired-end sequencing works, which is also known as short-read sequencing. So humans have a genome, and I'm just using this picture because it's colourful, but genomes are long and thin, so make it long and thin. We have two copies of most of our genomes, so I'm just going to show you two copies there. And we have variants, so I'm going to add these coloured lines to show to represent variants. And when we receive a DNA sample from a patient, what we get is lots and lots of copies of this DNA. And one of the first things we do is we break that DNA up into lots of little bits using fragmentation. And so we end up with a test tube with lots of fragments of DNA all mixed together. And then what we do is a size selection. So we remove all of the DNA fragments that are too big and all of the ones that are too small. And what we're left with is the ones that are about the right size to sequence. If we're doing targeting, we then add a load of probes which stick to the parts of the DNA that we actually do want to sequence. We then wash away all the rest of the DNA that we don't want to sequence, and what we're left with is the ones that we do. We then remove those probes and take them away. So now we have a test tube with fragments of DNA that are about the right size and we want to sequence. We don't know what's in them, so I'm just going to grade them out so that that shows that we don't know what that DNA is. But we put it in the sequencing machine and it sequences from one end and then it sequences from the other end. At this point we don't know how large each fragment is, so I'm just going to draw a line in between those two ends to show that. But then what we do is we take the data from the sequencing machine and we look at the reference genome, and one by one we take each read and we work out where that fits on the reference genome and just line them up against that. So that's a piece of software that does this called a liner. And as this goes on, we build up this picture of where the variants are, because you can see where the coloured lines are, that represents where the DNA in the read is different from the DNA in the reference genome. Once we've done that, we don't generally want to see where the DNA is the same as the reference genome, so let's just fade that out. And here we have a picture of where the variants are after doing the sequencing. So this is how paired-end sequencing is able to find the variants in a patient.